The overall goal of this procedure is to prepare aligned, shape-responsive liquid crystal elastomers and nanocomposites.
So liquid crystal elastomers are stimuli-responsive materials that change shape in response to stimuli in the environment, and they can be used in a variety of applications, such as tissue engineering, where we know cells respond to stresses and strains in the environment. In this procedure that we're going to demonstrate, we rely on materials that are low cost. The method we demonstrate is reliable, and it can be implemented in any lab without the need for expensive or specialized equipment.
These materials can be applied to the development of cardiac cell sheets, where we know that stresses and strains can affect the differentiation, the maturation, and the function of cardiac muscle cells.
Visual demonstration of this procedure is important because the process can be difficult to learn, and the sample is especially delicate when we're hanging, loading, and cross-linking it. So demonstrating the procedure will be Hojin Kim, Bohan, and Huiying Chen, students from my laboratory and the Jacot laboratory.
Begin by combining 166.23 milligrams of a reactive mesogen, 40 milligrams of siloxane, and 12.8 milligrams of crosslinker with 0.6 milliliters of anhydrous toluene in a small vial charged with a stirring bar. Stir the solution at 35 degrees Celsius for 25 minutes for the reagents to completely dissolve. In a separate vial, prepare a 1 weight % solution of a platinum catalyst in dichloromethane. Then, add 30 microliters of the